K2P channel gating, mechanisms revealed by structures of TREC2 and a complex with Prozac. Potassium channels are membrane protein structures that selectively allow potassium ions to cross the cell membrane dictated by the concentration gradient. Potassium channels, such as the two-pore domain potassium channel or K2P, are the main components responsible for maintaining cell potential in the vast majority of cells. They are critically important for excitable cells, such as neurons, in establishing the resting membrane potential needed to fire. A type of K2P, the TREC2 K2P, has been shown to contain a gating mechanism that regulates activity of the ion channel, and as such is an important target for clinical research, particularly in cardiovascular diseases and neurological disorders. Several physical factors have been shown to activate this gate, such as the mechanical stretch of the cell membrane, the cell potential, temperature, natural ligands such as the polyunsaturated fatty acid arachidonic acid, and the pH gradient over the cell membrane. However, these gates can also be manipulated and inhibited through various pharmaceuticals, such as anesthetics, neurological drugs, and antidepressants. In particular, the active metabolite norfluoxetine of fluoxetine, better known as Prozac, has been shown to act as a selective serotonin reuptake inhibitor by inhibiting particular TREC K2P channels, and is a common antidepressant used to treat many types of depression and anxiety disorders. The mechanisms by which the K2P channels sense the numerous stimuli are poorly understood. Therefore, we hypothesized that solving the crystal structure of the TREC channel in complex with activators and inhibitors would give a good indication of the mechanisms by which the protein structure operates. The crystal structure of the human TREC2 structure was solved for two conformations. The channel consists of two copies of the TREC2 protein that forms a dimer shown in different colors. The difference in the two conformations is localized to the bottom intracellular part of the channel. In this part, there are 4 helices on each dimer that are shown to be in either a down state, closer to the cytoplasm, or in an up state that's closer to the membrane. When in the downward state, the structure reveals a window inside of the channel below the potassium selectivity filter that is closed by the upward movement of the helices, in particular the M4 helix, which moves significantly. When the M4 helix moves from the downward state to the upward state, it places its side chain residues to block the window. Bromine maps were generated, and it revealed that brominated fluoxetine accumulated within the down conformation's window. This confirms the state-dependent blocking of fluoxetine, or norfluoxetine, by its binding site within the window. This is further supported when known activators such as mechanical membrane stretching and the addition of arachidonic acid led to the subsequent reduced inhibition of the TREC2 by norfluoxetine due to the induced upstate and obstruction of the window. Additionally, it was shown that norfluoxetine greatly slows the membrane stretch activation and as such plays a role in preventing the conversion between the states. In order to form a fully conductive channel, all 4 potassium sites must be occupied. Hence, this could explain the decreased activity of TREC2 in the down state that only contains the electron density to support 3 ions. Therefore, it may be in a non-conductive state, compared to the up state, which has been shown to have an electron density capable of supporting 4 ions. These studies allow us to define a gating mechanism that converts the TREC2 K2P between conformations. The conformations observed included a low-activity down conformation that is stabilized by norfluoxetine binding in the open windows, and a high-activity up conformation that is unaffected by norfluoxetine due to the closed window that is stabilized by membrane stretching or arachidonic acid. The results clearly demonstrate how the movement of the cytoplasmic helices create a regulatory mechanism for K2P channel gating using numerous stimuli and how the mechanism of inhibition of TRK2 by Prozac is state-dependent.